Testing 1, 2.
Hang on a second. I'm just going to use this. You watch everything, man. Well, it's just because I didn't have it plugged in. We'll be there in just a second. I bet you. That's it. We're there. Yep, that's me. All right. If everyone would take a seat, please. Come on in. We have some reserved seats up here and some other seats in the back. Greg, come on up. All right. Hi, everyone. I'm Randy Blakely. I'm the Executive Director of Stiles Nicholson Brain Institute. It's my pleasure to have you here in the Institute building. It's been quite an evening already. We've honored our guest speaker, Professor John Hardy, with an award for a New Horizons Award for Brain Science and Health, and also to Mr. David Nicholson for his enduring community investments in education and science. Now we're going to tell you a little bit about the program that you're here to witness. This is part of our annual Brainy Days series of public lectures. We have a fantastic speaker, Dr. Hardy, who's made seminal contributions to the field of Alzheimer's disease. We're very lucky to have him over today from University College London, but I won't steal the rest of his introduction from Corinne Lesmasis, but not yet, sit down. I have many more hours to speak. No. But because it's Brainy Days, I'd like to introduce you to Dr. Nicole Baggins, who heads our Brainy Days program and has worked with me tirelessly on many community education programs. It's really the spirit behind this whole endeavor. Nicole?
Good evening, everyone. Welcome to Brainy Days 2025. So as Randy mentioned, I direct our community programs, and Brainy Days is just one of them. Brainy Days is a month-long celebration of neuroscience that we hold annually. We have fun activities and talks for the public, and including this year's distinguished guest, Dr. John Hardy. I hope that you will check out our entire lineup. Our next speaker coming up actually will be again in this very room next week, Tuesday, March 18th. This is Dr. Michael Burton. Dr. Burton will be here for a talk and a reception, and he's going to talk about how alcohol and the immune system can affect pain sensitivity. Hope you'll join us for that. If you want to get out and about, please join us at the Cox Science Center in West Palm Beach on March 15th. That's this coming Saturday. We hope to see you there. That's a really fun, one of our favorite events of the year, where we have a lot of brainy, fun activities for kids of all ages. And we'll wrap up Brainy Days 2025 this year with Dr. Andy Newberg, and he will be at the Mandel Public Library in West Palm Beach on Wednesday, March 26th. He'll join us for a talk and a reception and a book signing following that. And he will discuss the relationship between brain function and religious and spiritual experiences. So that should be a very interesting talk as well. All of you can pick up our postcard and check us out. I hope to see you all again at some of our other events this month. You can also check out that little QR code on your phone if that's more helpful. But now I would like to take this opportunity to thank our very generous sponsor of Brainy Days, Palm Health Foundation, and introduce Pat McNamara, President and CEO of Palm Health Foundation.
That's right. I'm locked in. Good evening, everybody. Pat McNamara. Palm Health Foundation is proud to have sponsored Brainy Days over the past many years, and to see the caliber of the folks that you guys continue to bring is just astonishing. So thank you for continuing to do such great work and delivering on it. I just wanted to share about something that we have recently formed, and that is a Brain Coast Alliance. Given the amazing neuroscience assets that we have right here in our backyard on the Jupiter campus, we are joining forces basically to advance brain health and brain science in South Florida. So going all the way from St. Lucie County down to Miami is the vision, to be able to harness the amazing brain power that we have locally, and to really advance engaging the community and fostering innovation for what ails us most. So thank you very much. You'll hear a lot more about the Brain Coast soon.
So today we are also celebrating the launch of the David and Lynn Nicholson Center for Neurodegenerative Disease Research. We have recruited a fantastic scientist, Corinne Las Mesas, to lead the center, and we've recruited several faculty that work in Parkinson's disease, on Alzheimer's disease, and on Huntington's disease, and even brain cancer. So we have quite an exciting build-up now, and there's just more to come. So maybe you haven't seen nothing yet, as they say. So Dr. Las Mesas did her early work in France. She's French, and she has an extraordinary pedigree of working on neurodegenerative diseases, beginning with, at that time, a little-known disease called mad cow disease. You remember mad cow disease? Well, at one point, it was thought to just be restricted to cows. But then we saw a move, like we're worried about right now with avian flu, right? And what Dr. Las Mesas found was, in fact, that this was moving to humans, and the exact molecular mechanism by which that was happening. That was an astounding discovery in its time. And she's followed that trail forward, and fortunately, it has brought her here. She's now professor of clinical neurosciences in the College of Medicine at FAU. I've mentioned her discovery of this transmission via a protein that is now known to be a prion protein. And now we think that this prion protein, which can self-replicate and extend itself throughout the brain, is a strong model for how toxic proteins in the brain can lead to disorders like Parkinson's disease and even Alzheimer's disease. She's been an advisor to the World Health Organization, the FDA, and the USDA. She's a PhD as well as a veterinarian, which helps when you're trying to look at diseases moving from a cow to a human. And she also takes her work to a translational level in founding and working with biotech companies to take her discoveries and turn them into things to help people. And so that company, Vova-Ida Therapeutics, will actually take roost here in its formative stages here in the building around us so that our trainees and other scientists can take advantage of the technologies and the understanding that they will bring and teach us how to take our discoveries to market. So without any further ado, I would like to bring Corinne up, who will introduce our speaker.
Thank you very much, Randy, for this very, very kind introduction of myself, but I'm not going to be the speaker tonight. Our speaker is Sir Professor Dr. John Hardy, who is coming from London to talk to us tonight. John Hardy started his career as a neurochemist. In 1995, he retrained as in molecular genetics. Back in time, 1991, John Hardy discovered that a point mutation in the amyloid precursor protein, APP, caused Alzheimer's disease in a British family. This was the landmark discovery that led to formulate the amyloid cascade hypothesis, stipulating that clumps of a protein called A-beta initiates a series of toxic events for the brain that eventually leads to neuronal loss and dementia. Fast forward, this discovery led to the only 2 FDA-approved treatments that interfere with disease progression in Alzheimer's disease. He identified also tau mutations causing frontotemporal dementia and discovered that the triplication of the alpha-synuclein gene causes Parkinson's disease. He was awarded multiple honors for his work in Alzheimer's research and neurodegenerative diseases. He's the founder of the International Parkinson's Disease Collaborative Network and initiator of the open access databases Brainiac and Mouseac Genetics. In 2021, he was awarded a knighthood in the United Kingdom's New Year honors for services to human health in improving understanding of dementia and neurodegenerative diseases. I'm extremely honored that John accepted to come tonight and give us a presentation and explain all about these diseases to us. Thank you very much, John, for being here.
First up, while we're just doing the computing, I'd just like to say what an honor it's been and how well I've been treated here on the Brain. Is it Brain Coast? Brainy Coast. It's been. I worked in Jacksonville at the Mayo Clinic until 2001. I left, and it's been so. I've only been back once before. Since that time, and it's been such a pleasure to walk on boardwalks, see the wildlife, and just also feel the enthusiasm here. It's been a real pleasure. This trip has been a real pleasure for me, and I'd just like to say thank you for looking after me so well. So, let's, yeah. I hope like I said, I always am a bit worried that my emails are on and I'll show something inappropriate. So let's, I think let's, anyway. I'm going to start by showing a video which I think puts everything into perspective.
Quest for medical treatments and cures isn't just about slides and microscopes and what can you do about the symptoms. It's about families, it's about individuals, it's about stories of people who suddenly begin to lose themselves in a way that's frightening for them and terribly painful for those who love them.
This is about the first family we worked on.
A lot of people would say that Alzheimer's disease research, to some extent, started with Carol's family.
All of them were between the ages of 54 and 58. And it was at that stage that our life changed, really.
We put an advert in 1985, 1986 in the Alzheimer's Society newsletter because we were interested in studying the genetics of Alzheimer's disease. And the very first letter I got was from Carol Jennings, and she had drawn her family tree, described her family tree.
The breakthrough itself came in 1991. We got a letter in January telling us they had identified a gene that they believed causes Alzheimer's disease.
When we found the mutations in amyloid in Carol's family, we knew that that is where the disease started. It's been a dominant feature of all work on Alzheimer's disease.
This is at the Clinical Research Centre at Queen Square.
Carol's family has what's called autosomal dominant Alzheimer's disease, and that means that if your father or mother has the disease, you have a 50% chance of inheriting the gene, and if you get the gene, you're going to get the disease at a very similar age, probably, to your parent.
She's a shell of the woman she once was, but she's still my wife, so she's still part of who I am. We may not be able to change the cards that life deals with us, but we can change the game we play with the cards we've got so that future generations don't have to carry the same pain.
Yeah, she really was a force of nature, and she died. She's actually within about a month of my own age. So, you know, it was very difficult. I mean, of course, nothing really, but very difficult for me to see her go off. I mean, God knows what it must have been like for her husband, and you saw her son there, who's now himself at 50% risk. Where you saw her was at the Clinical Research Centre at Queen Square, UCL, which is where I work. I'm not a clinician, so she was being looked after and going on clinical trials with some of my clinical colleagues. She was supporting research at Queen Square in that way. What I didn't know until I attended her funeral recently, I attended her funeral, it's now about 6 months ago, was that she was also in her hometown of Nottingham, a fundraiser for a local charity which was putting through PhD students to work on Alzheimer's disease at Nottingham University. And the other thing I did know about was there's a rare disease, a rarer disease called frontotemporal dementia, and there's a national society for frontal temporal dementia. It's quite rare, so families rarely meet each other because they're so sparse. And she became the national volunteer organizer for that charity. And every year we would have a meeting, and she would do all the boring stuff. Like, she would help the families get somewhere to stay, so they, find them cheap hotels to stay in. She would be the person pouring the cup of tea outside the lecture theatre. And she would be the person people who were perhaps shy of medical medical people and researchers, they could go and ask her a question because she had been through pretty much the same things as they had been through. So she was a friendly face. Really, just a remarkable woman. Yeah, just a remarkable woman. And her husband, as you saw there, is also a remarkable guy. Just a pillar of the community, was a local vicar. And, you know, and as a local vicar, he was helping homeless. I mean, they were just really the salt of the earth, I don't know if you use that expression here, but they were the salt of the earth. And frankly, this research would not have started without them. And of course, there are other families like that. They are quite rare, but I think, as I hope I convince you, they've been really informative in terms of how we understand the disease. And I should say her son is now also going through clinical trials at Queen Square, UCL. So, you know, that's something that the family connection continues. Okay, let's get rid of this and I'll go to the slides now. You know, as the video said, this video was made about, let's get rid of that. There we go. So, now I will just say that once I was giving a talk and my sister contacted me while I was while I was giving the talk, and let me just say, I wasn't happy, and when everybody in the audience heard exactly what she had to say to me. Anyway, so this is fine as a view. This is fine. Where are we up to? I think, you know, I got that prize. This was a video from about 5 years ago, and when I got that, I have to say, of course, it was great, but I also felt, you know, we've understood a lot about the disease, but so far it hasn't done any good. I mean, you know, I think we had developed an understanding of the disease mechanisms, but actually in terms of the benefits to patients, they had not come then. And so it was actually a considerable relief, and I'm going to talk about the treatments that have come. It was actually a considerable relief to me, more than anything, that when the treatments based upon the amyloid findings started to come about in the last couple of years, it made me feel that they have this thing called imposter syndrome. You know, I felt less of an imposter because it was actually starting to yield benefits. And that's where I'll take this tonight. So this is the pathology of Alzheimer's disease. Top left, you have a section through the brain. Down here, you can see the enlarged ventricle. I'll try and, well, yeah. Well, you can see the enlarged ventricle. Sorry, I can't, I'm not showing, but it doesn't matter. You can see it anyway. Top right, you can see a cross-section along those red lines. And what I want you to look at there is down here at the bottom here, you can see that's the hippocampus. And that is the part of the brain, really, which is centrally involved in making new memories. And that's really shrunken in an Alzheimer case. And that's important because that fits with the clinical features. Often the first problem that somebody with Alzheimer's disease has is they find it difficult to make new memories. They can remember what happened to them in the 1930s and 1940s. They can remember those with a lot of clarity, but they can't remember, you know, whether they bought a pint of milk this morning. They're not making new memories, and that's because they've lost a lot of their hippocampus. So that's top right. Bottom left, you have a section through the through the cortex, stained, and these are tangles made up of the tau protein. These are about 1/10 millimeter across. These are stained with amyloid. These are the plaques of Alzheimer's disease. And here you have a blood vessel, and you have the same amyloid around the blood vessel. This has been, to some extent, the forgotten pathology of Alzheimer's disease, but that forgetting, has come back to bite us, as I'll talk about towards the end. This is amyloid 2, and I'll just mention that this is in a blood vessel. This amyloid is not in the brain, it's in the blood vessel, so it's not behind the blood-brain barrier, and that's an important factor. Now, of course, what this is a complex pathology, and when we those in the field saw all this pathology, we didn't know which came first and which came second and what the order of that pathology was. And there were lots of different ideas. Some people thought the plaques came first, some people thought the tangles came first, some people thought neither were important and they were both the tombstones of dying neurons. There was, and you'd go to a meeting, and I have to say, I started at this time in the 1970s, and the meetings would be typically of about 50 people, and out of the 50 people, there'd be about 20 different ideas about the disease. There was no consensus at all, and very few researchers. And at this time, in 1983, Jim Gusella in Boston published a paper on Huntington's disease. He and his group, Nancy Wexler and others, had collected a huge family with Huntington's disease from Venezuela. Incidentally, just to say something politically incorrect, this shows how important it is to work on populations other than North Americans, because this Huntington's breakthrough came from Venezuela. I think it would be very difficult to get a grant to go to Venezuela now, just by the way. But anyway, they found they found when they did this, they looked genetically and realized that the gene was on chromosome 4. And I was in Newcastle at the time, and I read this paper. I was doing neurochemistry and pathology, and I was studying the pathology of the disease. And I read this paper and thought, how cool is this? This is a way of understanding how the disease starts. If you can understand how the disease starts, and you can see from this pathology how it ends, perhaps you can, if you like, draw a metabolic line between how it starts and how it finishes and intervene in some way. That was my simple-minded idea. You know, I'm glad I did it. And so I was lucky enough eventually to get a job in the genetics department, and with funding from the MRC and the support of my colleagues, we started to do just that, to try and find the gene. So we advertised, as the film said, and Carol Jennings wrote the first letter. Her family, incidentally, was called Family 23, and it was called Family 23 for a really silly reason, and that is that I got letters in, and they came in quite quickly, and I only started to answer them when there was I've got 23, and I started at the top of the pile, and in fact, Carol's letter was the very first I received. But because there were 22 above it, she became Family 23. Anyway, that's really not relevant to anything, but just but anyway, so how I saw genetics was as a way of refereeing between all the different theories about the disease. I didn't see genetics, what we were doing, as a way of generating a new hypothesis. I thought, look, there's all these ideas, and genetics is an unbiased way of finding out which of these ideas was the right one. And I just want to point out this paper from the top left, from George Glenner from 1984. George Glenner was a pathologist, very nice guy, actually. I got on with him very well. And he had been isolating amyloid to get the protein sequence. And actually, he had started by isolating amyloid from the blood vessels, because that's a simpler tissue and he could get a purer and simpler prep. And he got a short sequence of about 20 amino acids from that protein, from the protein sequencing. And he did that in a typical case of Alzheimer's disease first, and then the next thing he did in this particular paper is people with Down syndrome inevitably develop the pathology of Alzheimer's disease. So he did the same thing with a case of Down syndrome and got the same sequence. And so he wrote in this paper, this is the first chemical evidence of a relationship between Down syndrome and Alzheimer's disease. It suggests Down syndrome is a predictable model for Alzheimer's disease. The naming has changed. Assuming the amyloid protein is a human gene product, it suggests the genetic defect in Alzheimer's disease is on chromosome 21. People with Down syndrome have got 3 copies instead of 2 copies of chromosome 21. So he clearly, when he wrote this, thought these people with Down syndrome are getting Alzheimer's disease because they're making too much amyloid, because they've got an extra copy of the gene. So really this is the amyloid hypothesis. And Colin Masters, working then at NIH with Konrad Bayreuther in Germany, got pretty much the same sequence by isolating plaque cores, and he also clearly. So these researchers obviously thought that amyloid was where it started, and I felt that when we did our work, I felt that genetics was the referee, and I thought that the referee called it, and the amyloid was how it started. This is the family that Carol brought. So Carol's father was one of the black triangles at the bottom here, and she had drawn the family tree, which showed, of course, her father and her uncles and aunts. 5 of the 10 of them had disease. Then her grandfather had the disease, and then her double cousins also, one of the 3 double cousins had also got it, and then the great-grandfather at the very top is where it had started. That's the family tree that she drew, and what you're looking at there is the top half of chromosome 21. And it's a very simple experiment, although it took us a very long time, and what you can see that all of the affected members have got the entire chromosome region, but number 9 and number 11 here have got bits of the chromosome and are not sick, and so that localizes the disease gene between those 2 bits, and the amyloid APP gene is in the middle, and we sequenced it and found the mutations. That is the essence of how genetic analysis works. It's simple. I'll just say, in defense of it taking a long time, the human genome hadn't been sequenced at that time, so we were really wandering around in the dark because we didn't have the human genome sequence. We didn't have PCR. In fact, PCR, which of those of you who work in labs will know more than you want to know perhaps, it came into play just about at this time. And also we have made a very important incorrect assumption. And the incorrect assumption we made was that everybody with this form of Alzheimer's disease had a mutation in the same gene. So we thought that all the families with this early onset disease would have a mutation in the same gene. And that turned out to be incorrect. There are other genes, presenilin 1 and presenilin 2, which I'll talk about in a second. And so we were mixing families with different etiologies. And really the important thing that made us make the breakthrough, so to speak, is that we decided, instead of pooling data from families, to analyze each family separately. That was the key change that made us. Now that's the story here. This, for those of you who are interested in, when I was here in Florida especially, I used to watch a terrible TV program which was called Hollywood Behind the Headlines. And it was trash TV, I have to say. And what it was about nearly always was child stars who had gone off the rails and had terrible midlives and then had come, in other words, it was the dirt, basically. Well, this is Alzheimer's genetics, the story behind the headlines, and exactly all the mistakes we made. So that's, if those of you are interested in mistakes, so to speak, that's what that's around. So anyway, just by the way. So we found the mutation in this family. We found the similar mutations in other families, but most families did not have amyloid mutations. And we realized pretty much immediately that there were other families which didn't have amyloid mutations. That led us to race, all the different groups raced to find the other mutations. The race was run essentially by Peter Hislop, who was then in Toronto. He's now at Columbia. And he identified mutations in a gene, in a protein he called presenilin, well, two presenilin, presenilin 1 and presenilin 2, and that's drawn here. So there you have the structure top left of presenilin 1. It's well, for those of you who are not kind of used to seeing biological structures, you're looking at a membrane here, and this protein in blue is going through the membrane 9 times. And all the residues in red are the residues at which we and others have found in different families mutations. And in yellow, what you have there is the amyloid precursor protein in what is the active site of presenilin, So the key breakthrough, which was made by Bart de Stroeper, Dennis Selko, and Chris Youngkin, essentially, was to realize that presenilin was the enzyme largely responsible for producing amyloid from the amyloid precursor protein. So really that is strong evidence for the amyloid hypothesis. You have either the protein itself, the amyloid precursor protein in yellow, or the enzyme responsible for metabolizing it in blue. Mutations in either of those genes cause disease. Amyloid mutations generally, there's some exceptions, but amyloid mutations generally cause disease onset, like in Carol's family of the 50s. The presenilin families generally cause disease in the 30s and 40s. It's really dramatic. The youngest family we worked on had an age of onset of about 32. The youngest I've ever heard of is a Polish family where the age of onset was 18. So really just an incredible. And what these mutations all do, and this is a paper, actually this paper is from the Mayo Jacksonville when we were there, led by Steve Youngkin. What these mutations all do is they cause an amyloid, a less soluble amyloid, to be produced. So amyloid deposition becomes more likely because the amyloid is a bit less soluble. I'll just take a very brief diversion, and that is that when we were collecting all these Alzheimer families, we also collected all the other families with different dementias. We collected a few prion families, which we gave to John Collins to work on, but we also had some frontotemporal dementia families where there is just tangle pathology, and in those families with just tangle, that tau, we found tau mutations. So we had now found amyloid and presenilin mutations in Alzheimer's disease and tau tangle mutations in the frontotemporal dementia where you just get tau pathology. And that allowed us to do a very simple experiment, and that is to cross mice with amyloid mutations with mice with tau mutations. And when we did that, what we saw was that we got more tangle deposition when we did the cross, which told us that amyloid was upstream of tau. It's like thinking of the the Mississippi River. You know, amyloid is upstream of tau in terms of the cascade, and that allowed us to draw this very what's going on? It's not going back. Whoops, now it's there. This simple, incredibly simple-minded diagram of the what's the pathologic cascade, which we drew, which we drew, and we drew this in 1998. Now, I'll just say, I think this is generally true, this simple diagram, saying that APP gives rise to amyloid. Amyloid is pushing tau dysfunction, which is leading to tangles and cell death. I think that is generally, I still think that is generally true. Notice, though, there are a lot of question marks here, and those question marks were there in 1998, and they're still there now. Well, that's not so good. And then the other thing is that we've drawn this here, and we imply this is a neuronal membrane. So what we're implying there is that amyloid outside a neuron is causing tangles inside a neuron. So the only cell on here, the only cell is a neuron. And I think that is wrong too. So really, and the thing I always say to students is if you pose what are reasonably simple questions, like implied by these question marks, and after, well, I said this a few years ago, after 20 years, you haven't answered any of the questions, maybe the questions are not right. Maybe the questions are misleading. And I think that is the case. And I think that what I said to you at the beginning of my motivation was also, has turned out not to be quite right. I had thought of this as a biochemical pathway, rather like glycolysis. That's what I had thought. But anybody who has done anything with glycolysis knows that if you change the sugar concentration, glycolysis changes within a second. Biochemical pathways operate over seconds. And yet this pathway takes over 20 years to lead to disease. And something that takes over 20 years is of its nature different from something that changes over seconds. And so that also, I think, is what's, well, it's obviously oversimplified, but part of the problem. And the fact that we only drew neurons, I think, is also part of the problem. I have to say, I think everybody fell into the same mistake for a long time. You know, those of you who worked on neurosciences, we basically behaved as if the only cells in the brain were neurons, and all the others were just support actors, like the crowd scenes in, you know, in a good old Hollywood movie. They just were running around. But actually, of course, that's not how it is. Then I think we're understanding that we have to study all these other cell types beside neurons as well. So we postulated the amyloid hypothesis, and I think that nearly everybody in the field was prepared to believe the amyloid hypothesis, so to speak, for the very few families with amyloid and presenilin mutations. And most people were prepared to believe that, but when it came to typical late onset disease, which of course is the vast majority, people thought it was too much to extrapolate from these very few unusual families to the disease in general. And so we got a lot of pushback about the hypothesis, quite understandably, from researchers who said, you've got no evidence that this is what's happening in late onset Alzheimer's disease, which of course is by far the major problem. So the push was on us and others, of course, to try and find out the risk factors for late onset disease. I won't go into the technology for this, but we used something called genome-wide association studies when the technology was invented in about 2005 to try and find risk genes for late onset disease in the general population. And this really transformed the research. And what this allowed us to do was to scan the rest of the genome for things, not things which cause disease, but things which increased our chances. We had expected to find things which directly affected amyloid production, like in early onset disease. And that is not what we found. Those of you who are good observers will notice I write obviously in italics there, because in fact, I think that in fact they were amyloid related, we just weren't smart enough to see it immediately. What happened when we found all the risk genes, and when we drew this diagram, there are about 40 of them, now there's about 80. What we found was that most of them were involved in the immune system. By that we largely mean microglia in the brain, and involved in, well, we write in this paper cholesterol metabolism, but I think we really mean lipid metabolism. That was entirely unexpected. We'd expected to get things which were involved directly in APP metabolism, and what we found is microglia with not even the right cell type and lipid metabolism. A complete surprise. And when we started to do sequencing, and we sequenced large numbers of cases when the technology allowed, and we found mutations in this gene TREM2, which was a microglial gene and completely, again, unexpected. So by both the genome-wide studies and by sequencing, they all pointed at microglia. The surprising thing to us is given in this little statement here, was that we found that when we were doing this genetic work, another group in my department were also looking at the mice we'd made which had amyloid deposits. And those mice had changes in gene expression, and we found to our surprise that when the mice deposited amyloid, the gene which was upregulated the most was the very same gene. So we had two independent findings. Genetics told us TREM2 was important in disease, and gene expression told us it was the most amyloid responsive gene in the CNS. And so we decided to use that and formalize it and look across the genome to see which other genes were upregulated by amyloid deposition. And that's shown in the top left here. TREM2 is right in the middle of there. It's the most right circle there, that's TREM2. TREM2 is the gene which is upregulated the most by amyloid deposition. All the other genes on that map are also upregulated, but the ones that are circled are also Alzheimer risk genes. And so this is telling us that actually what we're finding is that genetic variability in the response to amyloid deposition is important in determining your risk. It's giving us a general view about what's important in Alzheimer's disease, and that general view is that it's how well you react to your amyloid deposition. So, late onset typical Alzheimer's disease, the risk is determined, at least in part, by how well you deal with amyloid. And I had already said that in early onset disease, it's determined by how much amyloid you make. So all of this data really fits with the amyloid hypothesis, the overall etiology. Now moving on to treatments. Obviously, the implication is that if you could remove amyloid, you would be at least part of the way to treating the disease. And the first steps in this came really about by accident by this group led by Dale Schenk, and this is in 1999. Dale Schenk did two papers at this time. The first was that he injected mice with amyloid so that they raised antibodies to it, and the second was he used commercial antibodies and infused the mice with those infused antibodies. And what he found was that in these transgenic models of Alzheimer, of amyloid deposition, these antibodies seemed to reduce the pathology. It took us a while. we all made a mistake in interpreting these, and that mistake cost us a few years. And the mistake we made was that actually these early antibodies, they prevented amyloid deposition. So when you gave them, the mice didn't deposit anymore, but what they did not do is cause amyloid removal. So if amyloid was already there, that amyloid was not cleared. That was a distinction we didn't not, well, I shouldn't speak for the whole field, but it's certainly a distinction that I did not make. I thought, you know, these look as if they're stopping amyloid, and that was enough for me at the time. I think that was a mistake. That was, well, my interpretive mistake. I think others made the same mistake. The other thing is, just again, by the way, even in this early paper, which is 1999, they saw what we now call ARIA effect. So what happens when you give an antibody, well, to a mouse or a human, the first thing the antibody hits is the amyloid in the blood vessel. Remember I said that the amyloid in the blood vessel is outside the brain. So that amyloid gets high concentrations of antibody, and the antibody causes the amyloid removal, but you get an inflammatory response. And many of these blood vessels are fragile. They've lost part of the protective tissue around the blood vessel, and when you remove the amyloid, it can be a mess. It can cause hemorrhages, and that is what ARIA is. And that was seen even then in 1999. But that is almost certainly the reason. The antibody, the first thing it sees, and at high concentration, is the blood vessel amyloid. So there were a lot of failed clinical trials, and I think that there was several reasons they failed, but an important one was that distinction between stopping amyloid deposition and causing amyloid removal was one of them. Another problem was that the diagnostic accuracy rate in these early trials was only something of the order of 70 to 75%. And it's very difficult to do a clinical trial of an amyloid removing drug when only, 2/3 of your cases have got amyloid to remove, and the others have got other causes of their dementia. So you're really weighting the pitch against yourself with that. That was another reason. That's changed now in the more recent trials. So I have to say, and I kind of implied this when I was talking about, being disappointed earlier on when we had not made any difference to the patients. This paper from Bart de Strooper and Eric Karen came out about 3 years ago, and they made a prediction of what we needed to do to have a successful clinical trial. And I really recommend this to anybody who's really interested in this subject, because, everybody, there's a tendency in science to do what I think you call here in the States Monday morning quarterbacking. Everybody is smart afterwards. And this has the benefit, this review has the benefit of being smart beforehand. And that is an unusual thing. And what they said was that this is the model you need to have for a successful clinical trial. And so you've got time along here. You've got the amount of amyloid in the brain on this axis. You start your treatment here, you start to suck amyloid out of the brain, and then you run the clinical trial long enough that you start to see a clinical difference between the cases and control. They said that's what you need to do for a successful clinical trial. You need to remove amyloid, and then the trial needs to go on long enough after you've removed the amyloid for you to see clinical benefit. And that's turned out to be absolutely true. And I think it's so, these are the trials. I'll talk about gantenerumab. Well, lecanemab was the first unambiguously successful, and that's now FDA approved. Gantenerumab was unsuccessful, but it's coming back in a different form, and I'll talk about that. And then aducanumab was the first to get approval, but nobody was really happy with the approval process. Anybody who read the New York Times and as well as the scientific literature, maybe it's 3 or 4 years ago now, might remember the controversy. People resigned from FDA committees because they didn't think it should have been approved, and so on and so forth. But then lecanemab came, that was successful, everyone agrees about it. And these are the data. And so I showed you the theoretical data from the Karen-Nadstrupa review, and these are the real data from the trials. And you can see this is donanemab, this is the Eli Lilly trial here, here's lecanemab, this is so donanemab and lecanemab are both approved. Aducanumab had 2 trials, and this is why it's controversial. This lower one is the one that statistically passed, but the upper one, which you see doesn't remove amyloid quite as well, failed. And that's why the approval was controversial. I'll come back to the gantenerumab. You can see gantenerumab removes amyloid, but doesn't do it very effectively. And over the period of the trial, which was nearly 2.5 years, it did not get down all the way. And I'll come back to that. So this is the lecanemab data. This is the amyloid removal here. And all of the others are basically clinical scales showing that you're slowing the disease. So you're slowing the disease, but you're not stopping it. And that is something that we don't understand, first of all. I think that's important to say. And there's a lot of work on this now to try and understand this better. Does this reflect ongoing damage processes? For example, is the tau pathology, has that become self-propagating by the time the trial was started, for example? You know, are there other reasons for the continuing decline? We don't know. We just don't know. So there's plenty of work to be done on this. And I've shown the lecanemab data. The donanemab data is essentially superimposable on this. And the other point is that the trials were very well organized. Everybody had very careful analyses to check that they did have amyloid deposition, not like the earlier trial. And the administration is still cumbersome, intravenous every 2 weeks. And ARIA, this response of the blood vessels to antibody attack, so to speak, it was a real problem. I should say that the symptoms of ARIA are usually, well, most people who have it don't realize it. It's amyloid-related imaging abnormality. It's the radiologist who sees it, not necessarily the patient. And some of the patients aren't aware of it at all. Some have headaches, but very rarely it causes hemorrhages, especially for those who are also on TPA and that sort of drug. So, and when that happens, you get very few, but some fatalities. So it's difficult. And that means that you really have to monitor, they have to be monitored. It's an expensive drug already, but the whole giving of the drug involves very careful and expensive monitoring. I don't know how much it's costing here in the States. The estimate in the UK is that it's something like £50,000 per year per patient. And this is really about half of which is the drug cost and half of which is the monitoring, the administration and monitoring costs. Now it is getting better. We get the administration is being tested for subcutaneous now, and I think that will come into practice soon. That'll make a big difference. Both Eli Lilly and Eisai are testing whether people can be given drug holidays, so that when they knock their amyloid down, can they go off drug for a while before they go back onto drug, which of course would cut the cost and also cut the caregiver burden, which, you know, if you're taking people in for treatment every couple of weeks is quite considerable. So, you know, there are practical ways in which this is improving. The other thing I think is it's obviously going to be important, and maybe my next slide, is we need to get better at diagnosing the disease earlier. As geneticists, we're working on this. We can use genetics to predict with a reasonably high degree of accuracy an area under the curve of about 0.8, using all of the genetics to predict who's at high risk of getting disease. However, this is in Northern Europeans, white Northern Europeans. The predictive accuracy so far in other populations is much less than this. We've got an ongoing collaboration with Hong Kong to look at Chinese population, and the area under the curve in Caucasians is 0.8. The area under the curve in Hong Kong Chinese is about 0.6. So, and that's because the structure of the genome is different and the risk genes are different, so we're much worse. We have to do this in all the other populations besides white Northern Europeans. We've got quite a long way to go. That's one benefit. The other is the blood-based biomarkers. Blood-based biomarkers are they've really changed. The technologies for blood-based biomarkers, if you'd asked me 10 years ago, is it ever worth trying to look for blood biomarkers of Alzheimer's disease, I'd have said no, it's too far from the brain. But actually, now with these new technologies, it's really, that prediction was wrong. Now blood-based biomarkers in experimental settings work, and we can predict who's at really high risk based upon their blood chemistry right now. Not yet in clinical practice, but in experimental situations. So those things are getting better. And of course, if we can give the treatment earlier, we might be able to stop the disease more effectively, but we should be able to avoid the side effect of ARIA because there won't be much amyloid in the blood vessels. So pushing the diagnosis earlier is also a really big push at the moment. Now, finally, can we avoid ARIA? And this is where gantenerumab has a second life. Roche have started to tie the active site of their unsuccessful gantenerumab to a transferrin receptor, so they can get the active site of the antibody through the blood-brain barrier. Rather analogous to what we heard about the ultrasound, really, it's the same basic idea. Getting a drug into the brain parenchyma past the blood-brain barrier effectively. And this has gone through a very small phase 2 trial, but it seems to work. It clears gantenerumab, which was a slow clear of amyloid in the original trial, is a fast clearer of amyloid when it's tied to the transferrin receptor, and it also seems to avoid the ARIA side effect, though this is a very small trial, and we need to see, like all of these things, we need to see a phase 3 trial. So where are we up to? Well, I tell you where I think we're up to, and that is that I think it was 1908, the Wright brothers got onto that North Carolina beach and did a figure of 8 on basically a modified bicycle. And, you know, if you'd asked anybody, would that ever lead to commercial flights? You'd have said, no way. Well, of course, I flew from London yesterday, so, you know, within 10 years, there were England to France flights. So things, you know, once something has been achieved, it's easier to do it again and to do it better. So I think that over the next few years, there will be undoubtedly progress. That will still leave things to do. I think we still need to understand that residual decline. We still need to understand, you know, what's the pathological process, which mean that when you remove amyloid, you don't stop the disease. So there is plenty of things to do. And I guess that you guys, like us back in London, want to be part of that process. But there's plenty of things to do and plenty for all of us to get on with. But I think we're on the road now. Thanks very much. Yeah, questions would be great.
Yep. So we need the microphone. Yes. Do we have it? Okay. And I think we have. Great. And we have one more? Okay. Corinne, you want to take that one? Yeah. And we'll just cover the room as best we can. So I'll kick off questions. I know that there are going to be a number of them, and we'll stay here till, you know, you want us to leave.
Oh, please.
Yes. Jet lag is sort of sinking in. So in terms of risk, we know age is a risk, right? But so is sex. So women are at greater risk than men. And when you show the data, are those men and women?
Yes, it is men and women. And of course, you're right. Women are at greater risk of Alzheimer's disease. Men are at greater risk of Parkinson's disease. I don't understand it. We don't know why. Yeah, we don't know why. I mean,
in the trials, if you split them, do you find?
There's been some discussion about that, which I've seen, which has been rather bad-tempered. People have said that if you look at men, most of the people in the trial were women. If you look at men who have an E4 allele, I think it was, then they show no benefit. But that is what happens, of course, as you start to cut the numbers down.
Right.
Then it's you know, you can't really make firm conclusions. So I've seen an argument about that, but not really definitive data.
Got it. Let's walk around here. Find some hands. Let's see one.
Thank you very much. As a neuroradiologist, I see this class of patients that typically present with large lobar hemorrhages, typically elderly patients. They don't necessarily have dementia, although some might. And in the absence of other obvious causes, aneurysms, trauma, tumor, we typically label this as amyloid angiopathy. And I'm wondering, have you or anybody you know looked at these? Do they share histologic characteristics with people who actually do have Alzheimer's, where is it just affecting the blood vessels in those patients if they don't have dementia, or is it also affecting neurons and things like that?
I mean, one of the things I'll say is there's not been enough neuropathology done on patients like the ones you describe. So I don't think we really know definitively about that. I mean, certainly some of them, and I've been to case conferences back at Queen Square about this. Some of them have, if you like, incipient, you know, the early stages of Alzheimer pathology as well. But in general, I can't answer your question, really.
Thank you so much. It was amazing. I would love to know what you're personally working on right now.
And I'm not allowed to say staying awake, is that right? So actually the genetic prediction. The genetic prediction. The biggest project in our lab at the moment is trying to understand, obviously, you know, the whole field has developed all of these genes involved in risk, but what we can see is that what drives the speed of decline is different, at least, yeah, in part different from what drives the genetics of risk. So what genes influence rate of decline? I would say that's the biggest project we have. And At the moment, the first thing that comes out is the interferon pathway. I saw that on the slide. I always look